Hello everyone. Miss Hafsa Sarfaraz. I'm audible.
Walaikum salam. Yes, sir, you are audible.
Is my voice clear?
Yes, it's clear.
Okay, thank you so much to let me know and a warm welcome all of you in today's class. We will discuss miracles happening in the field of molecular biology and yeah, Miss Hafsa, please continue.
Assalamu alaikum and a very warm welcome to all of you in our sixth and hopefully the final class of our course, Advancements in Molecular Biology. And today with us is Mr. Ghulam Murtaza. He is going to speak on the topic of molecular biology techniques and basically, as he said, the miracles happening in molecular biology. So he is basically the CEO and Vice President of Vitanova International as Alliance for Sciences. Uh, Beside this, warmly welcome you all. And if you guys have any queries, you can write in the chat. We will surely take at the end of the session. So now I would like to hand over to Mr. Ghulam Murtaza. Over to you, sir. You can take start.
First of all, thank you so much, Ms. Hafsa Sarfaraz, Vice President with our International Alliance for Sciences, to invite me in advancement in molecular biology course 909. I really appreciate the core management team and organizing team to make that course happen at international level with international speakers as well. Anyone, please let me know my voice is clear to everyone. Please tell me in chat box. Okay, all right. So moving forward to that, in today's class, we will discuss molecular biology techniques. That's important uh, to study the gene, to study DNA at molecular level. Okay. First of all, we will discuss the DNA extraction. You can say it's very basic and very important technique in biotechnology. For example, we are doing genetic engineering. We are dealing with CRISPR-Cas, and each and every thing within the genetic engineering uh, for detection of gene. First we have to get the DNA. All right, then we will discuss like this DNA, how much express and how much it make protein. Okay, so in literature, in scientific literature, there are different protocols for DNA extractions. For DNA extractions, we extract DNA from different samples. From, for example, from, if I talk about in plant sciences domain or plant biotechnology, there are different protocols to extract DNA from plant sample, and there are different protocols to extract DNA from human samples. Okay, like blood, saliva, and bones. Yeah. So, but the principle of DNA extraction are almost same. Okay, principle are same, but protocols varies sample to sample. All right. Here, principle of DNA involves our key steps: disruption of cellular structure. For example, DNA, DNA present in the cell. So first step is to break the cell wall or cell membrane, so that the all the material in the cell come out from the cell. Okay. At the next step, separation of DNA. you know in the cell there are RNA, there are ribosome, proteins, many stuff into the cell, but we need DNA. So we will separate the DNA by the, yeah, we'll separate the DNA from other stuffs, debris, uh, binding DNA. Next principle is binding DNA. Attach DNA of interest to a purification matrix. So, third principle is we will purify the DNA. Purifications is third, you can say, principle. At the end, we will wash the DNA that that is dissolved within purification matrix. Okay, so purification matrix will be just like methanol, ethanol, any substance to which DNA can make bond. Okay, by these steps, after going on through these steps, we get a pure DNA. Okay, and then we can store it, and then we can uh, use that DNA for further process. For example, we have a transgenic plant. Okay, we have genetic engineered plant. For example, genetic engineered cotton crop. And after genetic engineering, we want to check that uh, how much uh, the DNA of interest expressed. For example, we put the gene of interest into the cotton plant, and we will check how much that gene of interest expressed in that plant. Okay, or then uh, then we will take plant sample, any leaf or any stem part. Preferably we take mostly leaf part. Okay, then we will extract DNA by following uh, these principles. Okay, as I already told, principles are same, but in literature there are different protocols to extract DNA from different sample. Okay, moving to forward, for example, we extract DNA from plant sample, and then we will move forward for further step. Further step is DNA quantification. I just wanna give a short brief. DNA quantification mean we are quantifying the DNA, like how much DNA and how much pure DNA we get from that sample, from given sample. Even that sample is human blood or plant leaves or any saliva, bones, any. So moving to next, if anyone have any question, you can note down your question. At the end we will discuss your question as well. We will make that session interactive. like in the form of panel discussion. And at the end we also discuss your queries, your problems that you face throughout the course. And in upcoming events we will overcome these challenges. At the next moment, after getting after quantification of DNA, then we will move towards polymerase chain reaction. Polymerase chain reaction is just like a you can say a miracle in molecular biology. Like we just put one sample, or you can say one piece of DNA into a thermocycler machine, and it make millions of copies of DNA. Okay, for example, we get DNA sample in nanogram, and that is very low amount, and we cannot make further experiment with that small amount. So we will then through PCR we will make multiple copies of DNA. Okay, so that we can proceed our further experiment with that that amount, that is enough. So polymerase chain reaction PCI is molecular technique to amplify extend and like maximize the DNA. So key points: PCR make millions of copies of specific segment of DNA, specific part of DNA. Okay, that part of DNA is our gene of interest that we put into a any organism, even in bacteria, even in plant, even in human. Okay, through genetic engineering, through CRISPR-Cas. Okay, work like DNA photocopy machine. You can say it's a photocopy machine. And why PCI important? How study gene easily, save time and sample material. Okay, I just want to give you a brief about key step in PCR. For example, uh, first step is DNA. Yeah, first step is DNA breakdown. Okay, and you can say it's make some time during the these all reactions are going on in the thermocycler machine. And second step is DNA amplification. Okay, through that step, you can say uh, nucleotides bind with uh, that, you can say nucleotide bind with single strand of DNA. Okay, for example, we have a double stranded DNA. It break down into two single stranded DNA. Okay, and then nucleotides go on and attached with these single stranded DNA and make double stranded DNA. Okay, and through that going on, like within minutes there are millions of copies made by these steps. First is uh, yeah DNA breakdown, then annealing, and then at the end of extension. Like that is extension is end step of PCR. Okay, polymerase chain reactions. So these are like basic concept of PCR. As a student of biotechnology, everyone read about it and take it during their coursework. It's just like a revision. But if you have any question in solid question that in your mind, we can clear it. Okay, and you can say throughout your domain every person learn new things. Okay, uh, during the PCR step, there are many problems scientists faces. For example, when we are working in lab and when we are taking coursework, there is a huge difference. Okay, and when we are working in the lab, we are making experiments, then real world problem solve. Okay, so application of PCR here: disease diagnosis, okay, detection of pathogens, okay, and research and gene analysis, forensic science, DNA fingerprinting, and I want to add some points: transgenic plant uh, detection as well, and gene expression studies. That is very important PCR step. Okay, advantage of PCR, like fast and accurate, highly sensitive, widely used in laboratories. Okay, as I am plant biotechnologist, so I use PCR in plant sample mostly. Okay, but we study different protocols for human samples as well, like these disease diagnosis system or forensic science. Okay, at the next moment, after PCR, when we get enough amount of our specific segment of DNA, then we will go towards gel electrophoresis. Okay, gel electrophoresis is the molecular technique to separate DNA, RNA, and proteins. Okay, on the base of their size, like uh, different DNA or different segments of DNA have different size, like these size are in kb or in different units. So on the base of sizes, we separate the DNA through that technique. Like we when we go for different interviews, on even on for job or for scholarships, these are basic questions. That, for example, if you are molecular biologist, you are medical student, medical researcher, and plant sciences researcher, and genetic engineer. So these are basic questions they can ask. Okay, like for what Java lactose is used we use. So key points: separate molecule on the base of size, use a gel, agarose, or polyacrylamide. Smaller molecule move faster than larger. It's a it's a common sense thing, like which is smaller and smarter things move fast, and large things move uh, takes time. Of course, so that technique work on that principle. Okay, gel electrophoresis is molecular technique to separate DNA RNA. How it works: sample are loaded into gel, as you can see into the structure, and yeah, you can like see into the that structure, and uh, here is a machine that is somehow different that I used. So there are one tray, and there are one flask, in which we make the gel in the form of liquid, and then we will like, put the liquid into the tray, and it will be solidify mean it will convert liquid to solid form. Okay, and it's take almost take 20 to uh, 20 minutes almost. So after that, after solidification, we use a material for solidification of the gel, uh, different different chemicals use. Okay, so After that, we put the sample into the gel. Okay, there are wells into the gel. We put sample into the wells of gel, and then we supply electric current, like it that machine have cathode and anode, like positive point to negative point. Okay, when we apply you can say supply 100 watt almost, then DNA have a negative charge, it will move towards positive charge. Okay, when we give supply electricity, it will move towards positive charge because it have negative charge. So on that principle, DNA move on the base of size. Those are smaller in size, they will move faster, and those are bigger in size, they will takes time, and they will move slowly. So in that structure, you can see easily the steps. Uh, Next are types of gel electrophoresis. Gel electrophoresis used for DNA, RNA, and commonly used separate large fragment. Okay, gel electrophoresis. Okay, polyacrylamide electrophoresis used for protein and small DNA fragments. Okay, polyacrylamide type electrophoresis mostly used for protein analysis, and gel electrophoresis used for DNA mostly. So DNA and RNA as well. Mostly in Pakistan, these facilities are present in Pakistani labs. In the next step, DNA sequencing, as we read, as you people take second lecture about next generation sequencing, that are next generation sequencing techniques. I hope you learn a lot from that course. DNA sequencing is a molecular technique used to determine exact order of nucleotide in the DNA molecule. For example, we have a DNA molecule of 1 meter. Okay, and in 1 meter there are sequence of codes. For example, ATGC, a different pairs. For example, first pair is A and T, next pair is T and A, third pair is G and C, and next pair is G and C again, and next pair is C and G. Like in this order, DNA is present in our cells. Okay, so DNA sequencing basic meaning is we are going to determine the order, like in which order codons or these nucleotides A, T, G, C are present into the gene. And key point, identify the sequence of bases, help to understand gene structure, provide detailed genetic information. As you know, T codon combine, and they are make a specific amino acid, and then specific amino acid make proteins. And in this like it's called central dogma. For example, a specific part of DNA, okay, then it make messenger RNA, and after that, that messenger RNA go on to transcription, and then messenger RNA go towards translation and make the proteins, and that proteins make different functions into any organism, even in human or in plants. Okay, or in animals. Okay, in this way, that is called central dogma step. DNA to RNA and RNA to proteins. Okay, and proteins to different functions performing, and like all these proteins and hormones that are regulating our body metabolism, that are uh, that are just because of DNA. And like you can say a DNA is how much important in our body. Like it's it's a control unit. It's controlling everything in a living organisms. Okay. At the next moment, we are discussing the DNA sequencing. Reveal information accurately help study normal and abnormal genes. Like through that course, we can check where are mutations occur. Like we can detect mutations also. Applications of sequencing, identification of genes, detection of genetic mutations, disease diagnosis, cancer research, and genomics, medical research, biotechnology. Okay? And advantage of high accuracy and essential in modern biology. Like DNA sequencing is a very advanced technique. Okay, right now in next generation sequencing, Miss Mariam Sood told you about the third and fourth generation sequencing that are nanopores, like very small devices that scientists built to check the sequencing order of ATGC into any sample of DNA. Okay. Uh, at the next moment, we will discuss transcriptomics. Transcriptomics study of RNA molecule. After DNA to then RNA, transcriptomic is study in detail studies of RNA molecules produced in cell at given time. Okay, like gene expression, you can say. Study gene expression as RNA level, show genes are active, RNA level change with conditions. Transcriptome is equal to complete set of RNA in cells. Like uh, for example, we discuss genome. Genome is complete set of DNA in a cell, and transcriptome is complete set of RNA in the cell. Okay, so the study of transcriptome is called transcriptomics. Okay, at the next moment, Transcom- transcriptomic is the study of RNA molecules produced in cell at given time. Like it's called gene expression. For example, how much RNA produced from DNA, RNA produced from DNA, and then it's depend upon RNA how much proteins will be produced. Okay, gene expression is called how much RNA produce proteins, mean transcription to translation steps. Miss Hafsa Sarfraz, please give a reminder. Those are leaving the class, that is not good thing, and you can give a reminder into the group, and yeah, there will be not attendance in this way. Okay. Here you can see patient sample, transcription profiling, biological insights, applications of transcriptomics. Okay. At the next moment, application gene regulations, studying disease like cancer, drug discovery, developmental biology, give a real time advantage of uh, transcript atomics real-time gene activity more informative than DNA alone. For example, we are not getting any result at DNA level, then we scientists make studies at RNA level. So for better results, okay. Next is RNA. RNA interference is a natural process used to silence specific genes. And that is very just like a miracles. It's knock down the genes, silence specific genes. For example, uh, some genes involved in cancer. Okay, uh, so through that step, RNA interference, we can knock down, we can stop these genes so that these genes cannot make further RNA and proteins that are responsible for cancer. Okay, types of RNA, siRNA and miRNA. As siRNA, we will discuss and last class previous Miss Janato Firdos. How RNAi work? Small RNA binds to target messenger RNA, and messenger RNA that is synthesized by DNA is destroyed. Okay, when small RNA uh, bind with messenger RNA that synthesized by DNA, and in this way that is destroyed, and uh, messenger RNA turned off. When messenger RNA turned off, then there will be no translation and there will be no protein synthesis. Okay. Applications: studying gene functions, disease and cancer research. Okay, we can also detect plant disease as well. Okay, at the next moment we'll discuss CRISPR. CRISPR is like a very advanced technique right now. CRISPR-Cas12 and 13 are going on, especially in Cambridge and Oxford universities. These techniques are scientists working on, and also in China, some universities. So scientists are going on modifications within CRISPR. Okay. Firstly, CRISPR discovered in USA by a microbiologist, Jennifer, and her colleague, one. Yeah, they discover CRISPR-Cas9 technique. Uh, Basically, the that phenomena is taken from a bacterial system, that Pseudomonas bacteria. And that bacteria, for example, they were studying that bacteria, Miss Jennifer, because she was a microbiologist and she was studying the Pseudomonas bacterial system. And when she observed like that system is just like a miracle. For example, when any virus attack on that bacteria, it have a enzymes, it have a Caesar type enzymes, Caesar type restrictions enzyme. When virus put their genome into that bacteria, it activate is restrictions enzyme are basically proteins, and that enzyme cut the viral genome, so that virus cannot be multiplied further. And then they discovered that CRISPR-Cas9 technique by following that Pseudomonas bacterial system. Okay, so as biomolecular biologist, as a biotechnologist, you must have knowledge of it, like where is it start. If you are mostly students are very interesting in that technique, so there are basic two components: guided RNA and Cas9 enzyme. And in upcoming techniques that are modified in CRISPR-Cas, like CRISPR-Cas12, CRISPR-Cas13. Okay, so there are CRISPR-Cas12 and CRISPR-Cas13 enzyme basically. Okay, and guided RNA, same thing. Next is CRISPR-Cas application. If I talk about CRISPR-Cas application in Pakistan are going on, but in plant plant sciences. Okay, not in on human side, because some ethical concerns. Okay, uh, some scientists have, for example, related to Islamic study, we should not destroy the natural things, like those are created by God. So uh, we can say that in nature, DNA modification and genetic engineering is going on in natural system. And you can take the example of Pseudomonas bacterial system. That is a natural thing, that bacteria using restrictions enzyme, like as a molecular seizure, to for his defensive system. Okay, so that is not a like that thing, uh, like if we are using that technology for the benefit of mankind, that's we should do that. Okay, so right now CRISPR is a very, you can say, very emerging technique, especially its scientists are using it in on human, like on human experiment in China, medical, Nanjing Medical University, as well in other research institutes. There are specific institutes on human research as well. uh, if we talk about like bioinformatics, that is computational biology basically start when human research projects start in 1990 to onward. And going on bioinformatics, just like this technique discovered by Miss Jennifer, and right now it's modifications into that system like CRISPR-Cas12 and 13. It's targeting RNA rather than DNA. Okay, so that system is mostly used in on plant sciences, on plant genomes. Okay, scientists are widely using that technique in the field of agriculture for crop improvements. I always, if I talk about the Vitano International Alliance for Sciences mission, that is integrated life sciences dealing. Okay, I always relate that thing. For example, recently we discussed different ideas. Uh, Plant sciences linked with medical health sciences. For example, you have better food, you have quality food, you have natural food system, and you might have tackle with different disease, even with cancer, even with many other disease. with natural food system. Okay, so in plant is natural commodity. Okay, and uh, Allah subhanahu wa ta'ala have different compounds, make compounds into plant systems as well, that are beneficial and that are uh, like, for example, in turmeric and in ginger, that are horticulture crops, and in that plants there are anti-cancer and antibacterial properties. Okay, so that obviously that's related to health. Okay, so as much there are many examples, not only this, many example. For example, plant-based medicines, plant-based food additives, plant-based cosmetics. Like we are getting many things from plants, and that is the thing, mostly people ignoring plant sciences field. But in Pakistan there is not research system, R&D system, research and development in the field of plant science. We are just stuck on breeding system to just improve the crops. If we go towards CRISPR-Cas system and advanced techniques, we can make a very beneficial products, biotechnological products from plant systems. Yeah. Uh, CRISPR-Cas, and next is blotting. There are three types of blotting: southern blotting, northern blotting, and western blotting. Southern blotting is a technique used to detect the DNA. Northern blotting is a technique used to detect the RNA. And western blotting is the technique used to detect the proteins, for example. Okay, there are basically three techniques which are used in molecular biology to detect. Okay, Miss Afsa, please. Miss Afsa, please go towards. Yeah, we were discussing blotting techniques. All right. So there are basically three types of blotting. Here we will discuss Western blotting. Western blotting basically used to detect the proteins. Okay, separate protein on the base of sizes using antibodies to identify protein, also called immunoblotting. Okay, because we are detecting proteins on the base of uh, antibodies, so that's called immunoblotting also. Okay, so basic steps in protein separation, transfer to membrane, and antibodies detection. In this system, Basically, we use gel electrophoresis system. Okay, same system, gel electrophoresis system. Just at the end, we we add the membrane system where protein patch up, and we apply antibodies and antigens, and through detection, uh, we then add probes as well that make light or fluorescence, and then we observe the gene protein gene expression basically. Okay, application studying protein expression, disease diagnosis, confirmation and test, research and biotechnology. Okay, advantage. Miss Safsa, please leave the slide. I am dealing it.
Yes, sir, sure.
At the next moment, we are dealing with RNA sequencing workflow. Okay, uh, just like a DNA sequencing, uh, that is RNA sequencing. Okay, in RNA sequencing, like probably related with DNA sequencing. For example, we are not getting results from DNA sequencing, then we go for uh, RNA sequencing. Okay, at RNA study levels, you can say transcriptomics level. Okay, RNA is converted into cDNA. Okay, cDNA mean single DNA. Okay, one strand of DNA. Okay, then cDNA is sequenced using next generation sequencing. Okay, same step, we just make one step add, that is making cDNA. Okay, cDNA mean make a single stranded DNA, that called RNA basically. Okay, produce million of short sequence reads. Okay, in this basically in sequencing, we also make amplify the DNA just like PCR. You can say in this step, in this technique, PCR system or PCR principle involves in sequencing technique. Okay, there are many machines, there are many others, you can say uh, electronic systems for sequencing of a genome. Okay, and transcriptome as well. Next is RNA interfere. As I already we already study. Next step is ELISA. ELISA mean enzyme linked immunosorbent assay. Okay, ELISA is basically a technique used in molecular biology to detect uh, proteins. It's also used for detection of proteins. Okay, uh, mean to say that is it's going used for detection of DNA. Okay, sorry, DNA, not DNA, detection of proteins. Okay, In ELISA, basically what's the ELISA steps? ELISA basically a machine in which valves are present. Okay, these are valves present. We use. Mhm. That are the principles of ELISA. Okay, ELISA mean uh, enzyme based enzyme linked immunosolvent assay. Okay, through that technique we detect the proteins, okay, in any sample. For example, we have a we have a sample of proteins in a tube, okay, and we separate proteins just like a DNA. We extract DNA like that. We also extract or purify proteins as well. Okay, then we detect how much proteins are present in that sample. Okay, and then we put that sample into ELISA machine. In ELISA machine we put the sample, and then we put some antibodies and antigen, and on the base of antibodies and antigen interactions we detect the proteins. Okay, and we detect the protein. We use probes basically, you can say you can say it's make uh, aluminous chemical. Okay, uh, when it interact it that you can say blue, uh, yellow, green, and for just like a star, that is pro. Okay, commonly used in clinical diagnosis, detect infections, hormones, biomarker, provide high sensitive and specificity. Okay, ELISA disease are used to diagnose disease like HIV, hepatitis, and COVID-19, helping early disease detection, suitable for large-scale screening. For example, in COVID-19, there are a lot of samples, so that technique is best to identify, uh, to check the sample for diagnosis purpose. Okay. Our next chapter is cloning. Gene cloning, a very, like you can say, very interesting technique, okay, in molecular biology. You can say it's a basic step in genetic engineering. For example, we are we are going to what the step of genetic engineering. So gene cloning is first step. Okay, clone mean we are making uh, same thing, copying, just you can say PCR. In PCR system we are making copies. Okay, and in cloning system we are making clones of any specific DNA. Okay, molecular cloning, gene insertion. Okay, basically in that step, molecular cloning involve insertion of a specific gene of interest into a vector. Common vector include plasmid. Okay, vector also called plasmid. Okay, vector basically single circular DNA that uh, that work as vector. You can say a carrier molecule that take a gene of interest and put into any system. even in bacteria or any system. Okay, and recombinant vector carries gene of interest. For example, when biotechnological product is insulin. Insulin basically produced from a bacteria. We clone the gene of interest that responsible to produce insulin. We put you can say we clone that gene of interest into that bacteria, and into that vector, and then put into bacteria, and then we multiply the bacteria. When bacteria will multiply, that vector will obviously will be multiplied, and then uh, that gene will produce proteins in the form of insulin. Basically, insulin will be the end product, and will be, you can say, uh, hormone. As recombinant vector are introduced into host cell replicate, produce multiple gene copies, and enable large scale DNA amplification. Okay, basically we're doing same thing here, just like a PCR. PCR, you can say a machine, you can say advanced uh, electric electronic thing, and you can say E. coli bacteria or natural PCR, natural PCR, you can say. Yeah. Molecular cloning, widely used in genetic engineering, help in protein production here. Insulin, important in research and biotechnology industry. Basically, uh, some branches of biotechnology, for example, green biotechnology refer to plant biotechnology or agriculture biotechnology. Gold biotechnology refer to bioinformatics, and red biotechnology related refer as health biotechnology, and blue biotechnology refer as uh, you can say marine biotechnology, like marine product, and that is very, you can say, interesting, and like very uh, like important in any country economic development. Like for example, in Pakistan, we have oceans, we have rivers, we have natural, Allah subhanahu wa ta'ala give us each and everything, but we are not using a positive way. We have oceans, and we can make wonderful products. As in European side, they are making wonderful product, and they are going towards entrepreneurship through biotechnology. But in Pakistan, there is no scope of biotechnology, especially in developing countries, in Bangladesh and in other countries like Nigeria, because they have not too much resources to make uh, like product, and there are many factors, political things as well, uh, where why we are not going towards to develop own product, and then go towards marketing and, yeah, entrepreneurships. Okay, so as a biotechnology or life sciences student, you must have a goal, future plannings. If you are a biotechnologist, you have to make at least one product within your life, for the benefit of mankind. At least that should be in your mind. You have to make proper planning how to achieve your goal. Okay, it's my sincere advice to all of you, as we are going to end that course. almost 100 plus students from 400 students. They join the classes and they make commitments. And I think if you are come here, you are learning things. You are learning from international scholars, and Allah will make opportunities. Allah will uh, give payback to your time that you are investing in your learning. Okay, and I really wish you good luck, all of you, in your future journey as well. Some are mostly from uh, life sciences domain. In our organization, we are mostly dealing uh, with the life sciences things as well. Okay, so stay here and be connected with the core team. And if you face any problem in your career development, you can discuss with us. Actually, through that courses, through that online opportunities, creating for the students to build their portfolio. And after that, we have the mission to support students to get high education scholarship around the globe to continue your higher studies. And you go for as a biotechnologist or as a life sciences student, you go towards a research field, and you have a long term future goals as well. For example, you are biotechnologist, you have must to make your product in master in PhD, and then you mm, make marketing it. Like, if you will work hard, uh, at least Allah will pay back. Okay, so okay, so uh, at the next moment, at the end, uh, yeah, I will provide the that's all slides that you need. And at the end, you can ask your question related to today's class. And you can also uh, take any question from previous classes. And you have if you have any queries, you can discuss here. Okay, we will sort out your problems in every condition. And really appreciate those are taking classes, those are learning, because you can say in arranging one class there are a lot of efforts in our teams, like inviting instructors and managing times and online training system. Each and everything take a much struggles. For example, in group there are 400 plus students, around about 300 plus students, but join only 48, 68, 98 roundabout. And I we always cooperate with the student with their genuine reason always. Okay, but in next step, we will just accommodate, we will support the student, those are dedicated, and those are really want to grow in their career, and we will support back them. We have organization will support our all students, those are affiliated with the Vitano. Uh, if I talk about the certifications, you have to make proper report. And after making the report, that we also share the sample into the group. According to sample, you have to make the report. and then you will submit your report with your names and each and everything through Google Form. Okay, and after that we will proceed your certifications internationally recognized. Yeah. It's depend upon for example, we have courses, we have internships, we have different other opportunities, even research projects. Is totally depend upon students interest, how much they are passionate, how much they are dedicated, and how much they are committed with their words. That's thing. How transcriptomics involve in drug discovery. Actually, for example, we are working on drug development. Okay, so mostly on initial steps, transcriptomics involved, but uh, transcriptomic have not much role in drug discovery. In you can say proteomics have too much role in drug discovery rather than transcriptomics. Okay, because there we are dealing uh, different uh, compounds, drug compounds that interact with proteins, protein inhibition, protein synthesis stops through the drug. Okay, for example, antibiotics work on that principle. Okay, antibiotics, for example, any proteins going on, and when we give the proteins, then infection stops. That basically uh, work on that principle. Yeah. Of course, we are actually our teams working day and night, of course, and we will make more opportunities for the dedicated student, those really want to grow, and we will support that to them in their career development. Okay, especially those belongs to life sciences. We are integrating life sciences domain with other domain, just like IT fields. You can say drug discovery, and other one is you can say uh, bioinformatics. Okay. The leaves of plant are saved in a Ziploc bag with uh, actually silica gel, uh, when we take plant samples, uh, for example, we have a disease plant leaves sample for DNA extractions. So silica gel not uh, say make the save to save the plant leaves for longer time. It can just save for short time to make the sample reliable for next procedure for DNA extractions, for DNA quantification, and then uh, PCR step, and next step gel electrophoresis, and southern blotting, northern blotting, western blotting, and then at the end DNA sequencing, and then bioinformatics, so on. But silica gel make sample safe for short time. Yes, we will work on it. We will make opportunities according to students' desire and according to the student opinion as well. Okay. If anyone have any question further regarding the today's class, previous classes, or any suggestion, if you have any idea, you can discuss here in chat box. We will include conclude that session within 15 minutes. Yeah. If anyone have any question further, want to discuss anything, if any one person go want to be live here, we can give access as well. Last date of report submission is 10th of January. 10th of January, we will announce into group as well. No, that report is just for you can say you to check your commitments. Okay, for evaluations, and after that we will provide certification. Certificate is approve, you can say evidence, you can say in your CV, in your CV profile, if you are writing anything, any conference, any seminar, any course, any internship, or any research project, anything you are mentioning, any volunteer work into your CV, into your portfolio, you must have the evidence of it. Okay, because science want evidence. Okay, science want results at least. Yes, of course. Our most instructors as practical experience of that field. Okay, but you cannot judge any person with one class. Okay, that is the my point. Actually, our platform is not bounded to anyone. Uh, for example, any learner from any domain who want to learn, he can join any course, any symposium, any opportunity to learn. But uh, at every stage, even in our cabinet, in our general council members, they are professor within our team. Okay, and according to their thoughts, they are still learning. If I am on a designation, I am serving students, I'm still learning, I am on learning stage, I am still serving, and according to our motto of that alliance is learn and serve. And still I am learning, and I am also serving. That's not big deal. Thank you so much for your suggestion as well. I really appreciate. But you are taking all classes, but all instructors was not same. Okay, uh, most minimum maximum one instructor take two classes. maybe, but uh, you cannot judge one instructor on the base of one class. Okay, it's depend upon everyone have different experience. I think we can make improvements. Yeah, no worries. Yes, we already at least student those are internationals. They have to make commitments before joining any any course, anything. Okay, because I have international exposure, uh, so it's not a big deal. International members here, they are managing their time. They are taking classes. I We have international students as well uh, in our course. They are taking the classes. Okay, they are taking the classes even from Bangladesh, from Rakesh, from Nigeria side, and they are managing their time. Okay, so you have to manage your time according to time schedule. If you face any problem in joining class, we are providing even recordings. We are making recording just like that. uh, it These will be available for future use. Even we recently make an internship. uh, Furthermore, thank you so much everyone for your participation, for joining today's class. And there will be one more class on tomorrow, may hopefully. And after that class you will work on completion your report, and after report submission via Google form we will start work on your certification, and within 1 to 2 days you will get your certifications. Okay, uh, within the group. And I really appreciate those really interested in learning process, because learning process is uh, not like stoppable. Like it's go throughout the life. Okay, so I again I really appreciate your efforts that you put into the that course. In future, we will make also collaboration opportunities for the life sciences domain students. Stay connected within the group. If you have to face any problem, you can reach out to me, Ms. Safsa Sarfaraz, Vice President, and other management team system. Yeah. So right now, if anyone have any question, you can you can write into chat box. One day after the course we will also open the privacy of group. You can make discussion there if you have any question queries. We will, yeah. uh, Dear Shazeb Ali. Okay, Shazeb Ali, actually, you can see into the group there. We always really cooperate with all members. Those have genuine reason, network issue, family issues, because it's a life and there are a lot of problems, especially in Pakistan. We always cooperate with all members. Okay, so we are also providing the online recording classes. You can take classes even when you are free. Okay, so how much we are cooperating, we are doing it. And those have zero attendance, those not take even only one class, they are not eligible for certifications. And those make good contribution throughout the course, soon they will get back their certifications. Right now we are going to end our class, and we are going to just, you can share your serial numbers at the end of class, and then we will include it. Thank you so much everyone to make that class interactive. I really appreciate your questions as well, your suggestions, your ideas. Really nice of you, and meet you soon back, and Allah Nigaban. Allah Hafiz.
Thank you so much, sir, for such an insightful session, and I'm really thankful to all the participants who are still here at the end of the session. I know it's too late, and but thank you so much, everyone, and special thanks to the speaker, Mr. Ghulam Murtaza, CEO and President of Vitanova International Alliance for Sciences. Thank you so much, sir, for being here today. And I really appreciate the participants and their participation in this session and their questions. And we will hopefully a class by tomorrow for related to drug designing and transcriptomics with Miss Adiba, and then we will finally conclude our course of advancements in molecular biology. So, till now we have done with the session. You all can leave with marking your attendance in the chat. Thank you so much, everyone.
Mr. Mansoor, am I audible to you? Am I
Yes, sir, you're
audible?
audible.
Okay,
Yes, sir, you can.
can I leave? You can further proceed it.
Yes, sir, you can leave. I will end the session.
Hopefully, you all had marked your attendance in the chat. And as usual, if you have any genuine reason, you can reach me out on WhatsApp so we can consider it there. So now, for now, we can end our session. Mr. Mansoor, you can end the class. Thank you so much.